Hello, welcome to today's webinar. We're going to be talking high-throughput genome editing with Dr. Kevin Roy from the Stanford Genome Technology Center. I'm Harry Singer and I'm super excited to see so many of you here today. It's being brought to you by my company, Singer Instruments. We've been providing lab automation solutions to the microbiology community for more than 40 years. It all started with classical yeast genetics, but now we're serving labs in different disciplines across a whole host of different organisms in over 60 countries. And as you'll hear shortly, Kevin's team have been using our colony picking robot PIXL to pioneer their innovative new CRISPR gene editing technology. It's called Majestic, and it looks like it's going to blow other library scale guide donor systems out of the water. It's pretty awesome. But that's not the only reason we're here today. In the last 12 months, Kevin's managed to pick an incredible 1 million colonies with PIXL, all with next to zero errors. And this webinar is partly to show you exactly how his team did that. Now, without further delay, it's time to welcome Kevin to introduce his amazing research along with some of the equally majestic robots that are making it happen. Over to you, Kevin.
Thank you, Harry, for that kind introduction. Again, my name is Kevin Roy. I'm a research scientist in the Department of Genetics at Stanford University School of Medicine and here at the Stanford Genome Technology Center. Here we develop technologies to dissect complex traits in genetics, and I'm pleased today to present our work developing a new genome editing system for dissecting complex traits using the yeast model system. So the first part of my talk, I will lay the foundation for yeast as a model to dissect complex traits. In the second part, I will describe a new genome editing system we built based on CRISPR that we call Majestic 3.0. And in the third part, I'll present how we use this system to dissect the causal variants underlying quantitative traits. So first, to motivate this work, we know in all natural populations that a major component of trait variation is due to genetic variance. And genome-wide association studies have uncovered thousands and thousands of links between genomic regions harboring genetic variation and different traits. And what's continuing to, these linkages are continuing to expand with the most recent numbers, eclipsing 700,000 lead SNP trait associations. Now, although these are associated with a particular SNP or lead variant marker in each region, in reality, each region harbors anywhere from dozens to thousands of genetic variants. And so in most cases, it's impossible to know what the causal variant and thus what the mechanism is underlying the trait for each locus. And so in order to tackle this problem and understand really the genetic architecture of each trait and each locus, the ideal system is to test each variant in isolation in high throughput studies in a systematic fashion. And so these can be broadly separated into three major categories that have been developed over the years. One of these is for looking at single nucleotide variants in regulatory regions. And for this, you can develop reporter assays where these are cloned into large fragments that harbors some element to dissect. One can approach variants that reside in regulatory regions in a more of an indirect fashion using CRISPR interference or activation screens to infer the function of a variant that resided at a target site and how that affects transcription. And ideally, the most relevant and powerful methods are to edit the endogenous variant at the locus in the genome using CRISPR methods. And so one of the reasons why we're most interested in single nucleotide changes are that these are the predominant source of phenotypic variation in natural populations. So across different species, we see different levels of variation depending on the size of the population. But the common theme is that the single nucleotide polymorphisms are present throughout. And so we can use model systems to uncover general principles that inform how complex traits are shaped by genetics. And so yeast has led the way in very many areas of both basic research and biotechnology. And so it was a pioneer system for systematic mapping of the genome and for developing functional genomics approaches. It was the first eukaryotic genome sequence, the first full collection of ORF deletions genome-wide, and then the use of those collections to infer gene function through barcode sequencing studies, first barcode hybridization to arrays, and then later on sequencing. It was the first organism to have a systematic double deletion analysis undertaken to understand how genes interact with each other. And it was also a pioneer organism for dissecting complex traits, with the first study to go from a trait down to the causal genes of that trait in a single study, as well as the first expression quantitative trait loci mapping. And as I mentioned, it's a crucial organism for the bioeconomy in very many sectors, including food, beverages, bioethanol, pharmaceuticals, and high-value small molecules. And so yeast is also an excellent model for dissecting the genotype-phenotype map in natural populations, but it was only until recently that we had a really global understanding of how yeast evolved out in nature and where the budding yeast that we all use in the lab actually came from. So one of the interesting findings of this study several years ago was that budding yeast originated in China and then spread throughout the world, in particular led by domestication, probably through human activities. Genome evolution in the wild isolates is primarily driven by the single nucleotide polymorphisms. Again, this is relevant when looking at yeast as a model, because this is what we see in other natural populations as well. And in line with that, most of these SNPs are present at very low frequencies. And the importance of this is that it's very difficult to functionally interrogate rare variants through association studies because they'll be present in so few individuals. So this really motivates the use of genome editing and functional assay technologies to look at these variants outside of association studies. And so in order to tackle this, we will now look at a method we developed for saturation editing of the genome. And when we talk about saturation editing, what we're referring to is the ability to engineer all possible single nucleotide variants across a genomic region. And so the way that these methods are employed is that you basically have a library of CRISPR guide RNAs. They start out as DNA, and they're linked to a template. In this case, it's a repair template that can be used by the cell with homology-directed repair. Now, what's powerful about this approach is that these guide donor sequences can be synthesized in large pooled format, so you can generate thousands of edits at a time. They need to be cloned into a proper plasmid backbone, and this presents an opportunity to barcode them so that every variant is tagged with a unique barcode. And then when these are transformed into cells, each cell will get a different guide donor plasmid. The guide will cut the target site with the Cas nuclease, and the donor will be serving as a template for homology-directed repair. And at the end, you have barcodes that you can sequence to interrogate the abundance of each variant in the pool. And you can do this across many different growth conditions to infer how the genetic variant impacts fitness. So these methods were developed in our lab as well as several others. And in order to appreciate how these approaches actually work, you first need to understand that even in the yeast system, which is well regarded for its efficiency of homology-directed repair, the rate of HDR with the donor, especially a circular plasmid donor, is insufficient for high-throughput library screens. And the reason for that is that there are alternate repair pathways that occur when a double-strand break happens. One of those are, as people well know, NHEJ indels. A major outcome, especially in the yeast system, is actually cell cycle arrest and eventually cell death. And furthermore, there's the potential for a recleavage of the target site after it's been edited, especially in the case of a single nucleotide variant, as these Cas nucleases have a mismatched tolerance that they've evolved, likely to have enabled targeting of viruses that accumulate variants in the regions of the guide RNA target. And then another related pathway would be the perfect repair of a target site, which is probably the most predominant pathway for many guides, as we know that editing takes quite a bit of time in many cases. And what's probably happening in these is that there is some level of perfect repair that's going on. All of these pathways are competing with the homology-directed repair with the donor. So what we developed was a method to enhance this process through recruiting the donor template to the target site using the FHA or fork head associated domain of FKH1. And we fuse this to a Lexase DNA binding domain. And then we can put Lexase sites on the double-stranded plasmid to recruit the donor. And we showed that this vastly enhances editing efficiency. So we wanted to compare this method with several other approaches that were employed to enhance HDR. So first, we start with a system that does not have any HDR enhancement. And then we can compare that to a system where a previous group inhibited NHEJ to enhance HDR. Another method was to use the Retron bacterial, it's an antiviral defense system. It's evolved similar to CRISPR, where it has a protein component. In this case, it's a reverse transcriptase, and an RNA component. And in this case, the RNA gets reverse transcribed into a DNA, and you can engineer these. A portion of these RNAs can be engineered to harbor any desired donor template sequence. And it's a very effective method for producing lots of donor template in the cell. A third method was in vivo plasmid assembly, developed by a different group several years back. And this was shown to enhance HDR, likely to presenting double-strand broken ends that the cell perceives as a substrate to enhance, to activate the DNA damage response prior to the CRISPR cut. And then we developed this method of donor recruitment. So we wanted to assay all of these in a single setting across a wide range of target sites. We chose 24 different targets randomly selected from a natural variant library, and we cloned them into plasmids suitable for each method. And we either looked at editing efficiency of each plasmid individually in a population of cells, or we pooled these plasmids and did an editing survival assay to look at how each edit competes with the other edits during the editing process. So the variant representation we can infer by sequencing the barcodes. And so what we found was that editing is a dynamic process, and that in the absence of any HDR enhancement approach, which we're calling wild type here, over the course of 18 generations of Cas9 induction, we can see the editing does go up, but then it peaks around 12 generations, and there's an accumulation of a large number of NHEJ indels across these target sites, with a mean editing efficiency here at 50%. With these other approaches to enhance HDR, what we can appreciate is that, first of all, the indels drop dramatically, and the editing efficiency with the donor DNA goes up as well. And they all perform quite well, with the FHA donor recruitment system performing the best. Then what we found was that systems that used constitutive editing and glucose generally had higher efficiencies straight off of the plate, of the agar plate. So the colonies that are washed off the agar plate are sequenced here at time 0, and then they're grown for an additional number of generations indicated here, with the optimal level being around 6 to 12 generations. And so what we found was that we could combine the donor recruitment system FHA with plasmid assembly to achieve superior results over either system alone. And so what you might notice is that even here, without any HDR enhancement approach, the editing was quite efficient in the case of glucose. But what we then found was that this comes at the cost of a dramatic difference in survival. So in this case, most of these edits have very poor survival when we if we look at a reference where there's no editing going on, so either with a no Cas9 control, we can see that the barcode levels are constant across this growth time course. And then we can also spike in a non-editing control to appreciate the toxicity of genome editing in the pooled screen. And so you can see that some guides shown here with a red line near the top, near the green non-editing control, or near the non-editing controls, indicating a low toxicity of editing, but the majority of them drop dramatically, with this y-axis representing the variant representation in a log scale. And so what we found was that some of these other methods, while they block the accumulation of NAGGA indels, they didn't do as much to improve survival. And so the skew that's observed is not improved. Whereas with the donor recruitment, we do observe a much better distribution of edits in the pool. And so what we found was that, again, by combining the plasmid assembly with the donor recruitment system, we get an improvement over either system alone. And these results show that roughly 6 to 12 generations of additional editing after the colony formation on the plate is the optimal level for editing. And so another feature of the Majestic system, which we have named for the long acronym of multiplex accurate genome editing with short trackable integrated cellular barcodes, is the barcoding process. So we take the barcode that's initially starting on the plasmid and we integrate that into the genome at a dedicated location. And so what that barcode is useful for is not just tracking the abundance of the variants in the pool, but we can also use that barcode to identify colonies that we isolate from these pools. And so we wanted to have an idea of what the genome-wide editing outcomes are for when we target natural variants, because when we're looking at the pooled assay, we're only able to use primers around the target site, and we're looking at a heterogeneous pool of clones that have each been edited independently. So here what we can do is look at clonal editing outcomes by plating for colonies and then using colony picking to generate arrays. And so we first did this with a standalone PIXL colony picker from Singer. And this required a technician to swap the plates out one by one. And so in this initial run, we picked around 12,000 colonies of a Majestic natural variant library. And this took us roughly 2.5 weeks of constant picking. So a technician every about 1 hour or so has to swap a plate out. And that enabled us to get a large number of arrayed colonies that we can then process further with a method we developed called recombinase directed indexing several years ago. And what we extended this method here to look at the Majestic barcodes that are integrated. And the way this method works is that the library that we've arrayed is an opposite mating type of the barcode array that we have. And in each genome, we have a LOXP site. And the barcoding array adjacent to this LOXP site is a barcode that is known for each position on the array. And in addition, it includes a Cree recombinase. In our Majestic edited library, we have a LOXP site, but on the other side of the LOXP site is where we deposit the barcode of interest. This barcode we don't initially know at first for each colony, but it corresponds to the variant we intended to edit. And so what we can do is, after mating, the chromosomes harboring these barcodes are in the same nucleus, and then we can induce Cree recombination. We use this ready process to identify colonies that have unique barcodes. We don't want to sample the same barcode over and over again. And we also use this to isolate sequence perfect guides and donors from the library, because these libraries have synthesis errors in them. And through the process of cloning and editing, you can enrich for some errors in the guide sequence. So at the end, we're able to use the robotic colony picking with the PIXL to further reduce this array into a consolidated collection where each variant is present with a unique barcode on the array. And what that allows us to do is access these colonies for further studies such as whole genome sequencing validation, as well as pooling them again for high quality liquid competitive growth, as well as agar phenotyping. And so what we wanted to first understand was what are the genome-wide editing outcomes. We know that at the target site, it looks like we're getting a very high efficiency with the system, but we wanted to confirm that we're not seeing unintended edits elsewhere in the genome. And so what we did was we took the colonies that we had arrayed, and we performed whole genome sequencing on a library of edits. These target all 16 chromosomes of yeast all across the chromosome, randomly selected from natural variants. And we did whole genome sequencing to look at editing outcomes, both at the target site and genome-wide. And so what we found was a surprise. We were able to edit most of the genome correctly most of the time. But it's in certain regions of the genome, we found that on-target structural variations were highly prevalent. And so what we were able to show in this study is that these are mainly restricted to particular regions of the genome, and they manifest in two different ways. One of them is through deletions, and these are typically occurring between tandem repeats of different levels of sequence homology. So it can be difficult to predict just by looking at a site whether or not it's going to undergo a deletion, because these repeats can be quite far away from each other, and they can have different levels of homology. And in the case of translocations, we found that these are primarily occurring near the telomeric ends, and that these are non-reciprocal in nature, such that one chromosome loses its subtelomeric region, and a second subtelomeric region from another chromosome is copied into that chromosome, so you get a duplication of that sequence. And so we found that these are driven largely by homologies around the target sites that either match each other in the case of deletions, or match other genomic regions in the case of the non-reciprocal translocations. So this allowed us to train a machine learning model that will take in this data set that we've generated of 1,875 editing outcomes, along with the sequence composition, both near the target site on either side, which we call the local repetitiveness, as well as the repetitiveness between the sequence around a target site and the rest of the genome, which we call the distal repetitiveness or global repetitiveness. In addition, we included the chromatin context of different chromatin marks near the target site, as well as sequence composition of the guide into this model. And so in the end, what the model is able to predict is the tendency of any edit in the genome to remain unedited, or to undergo some form of structural variant formation in the form of a deletion or a translocation. And so here are the complete set of predictions we have for chromosome 1 for all Cas9 targets. And so what you can see at the top are the predictions that any structural variant will occur when targeting along the chromosome. And these are enriched towards the telomeric ends, as you see, but also at various hotspots throughout the chromosome. And then we can divide these into either deletions or translocations. And so genome-wide, what we find is that roughly 4.8% of the genome has a relatively high risk for structural variant formation. And we define this as anything above the dotted line here, corresponding to a score SV of 0.15. And our complete predictions are available for anyone to view on the SCORE website, hosted here at the URL I'm showing. So we wanted to see if we could address these sites by employing the new genome editing system we had built. The earlier clones that we assayed were based only on the donor recruitment system. So we thought that perhaps by combining donor recruitment, donor amplification with the retron and plasmid assembly, we could address structural variant formation and potentially overcome it. So what we did was we combined the recruitment with Lexay FHA with an improved retron system that we were able to also recruit using the MS2 coat protein system, and also adapting the linearized plasmid assembly as well into this single approach. So we call this Majestic 3.0 because here we are combining 3 orthogonal methods for improving HDR. And so what we did is we took these structural variant loci and we transformed them, guide donors targeting each site, into a pool of cells. And here we're doing a pooled WGS assay to characterize editing at the target site, particularly looking for correct edits, unedited sites, or some form of structural variant. And so what you can see here at one site is that without any form of HDR enhancement, there is a complete loss of coverage near the target site. This is near the subtelomeric. This is in the subtelomeric region, so we're undergoing a non-reciprocal translocation here. When we add just the Retron system, we don't see much improvement. With just the donor recruitment system, we do now see quite a substantial improvement. But as we begin to combine the other components into the full Majestic 3.0 system, we're able to overcome structural variant formation at this site. And so what we did was we looked at a total of 8 sites with this approach. And what we find overall is that the Majestic 3.0 system performs the best in overcoming structural variant formation, and in some cases completely rescuing editing. But what we find is there is a relationship between the predicted strength of a structural variant formation and the ability to rescue that site. And so what we can see is that when the score of the machine learning model, the score SV is above 0.5, the level of rescue is quite low, less than 50%. And in the absence of any HDR, the editing at any of these sites is almost completely resulting in structural variant formation. And so what we've shown here is that we can enhance HDR with the donor template to overcome structural variants, cell death, and any TJ indels. In separate work that I didn't show, we also know that plasmids can integrate at target sites, and we've developed a system to recover correct edits of those through inducible plasmid cleavage. And an additional problem that I didn't discuss here that we've also overcome is how to reduce the recleavage of an edited SNP using high fidelity variants of Cas9. And so now we're taking the system that we've developed and applying it using robotics, including high throughput colony picking as well as high throughput phenotyping, to dissect many traits and how the variants at each locus contribute to that trait. And so what we did was we wanted to first be able to start with a sort of a solid data set where we knew we had variants in a locus that had an effect, but we didn't know which ones, and also a case where we would have some positive controls. And so what we did was we took a previous data set that had mapped roughly 5,000 QTLs across several dozen conditions and segregants. And many of these QTLs, in this case 112, could be fine mapped down to the gene level. And so we took these genes and wanted to further dissect them down to the variant level. And so we used either SP-Cas9 or LB-Cas12a in the context of Majestic 3.0 to target all of the variants in these regions. And so what we wanted to uncover were what are the functional variants actually mediating the trait. Are they non-coding, synonymous, or non-synonymous? What is the agreement between the QTL mapping and the causal variants that we identify? How prevalent are synonymous variants being causal? What is the relationship between effect size and allele frequency? And do most QTL, for example, have more than one causal variant? And do they operate in more than one environment? And so what we did here is we took a library where we had all of the variants that we could target, along with several controls to generate premature termination codons to knock out the function of the gene. And we are editing each variant with several guides when possible. So this results in a total library size of roughly 40,000 edits. And here we use the colony picking robotic system with the PIXL and the Spinnaker arm to array roughly 5 times the library size. So this is on the order of 200,000 colonies. And this is important because you expect some variation, of course, in the abundance level. So you need to pick well over the library size to isolate a majority of the desired library. And then at the end, we're able to consolidate a collection of the edited clones that we can then process for phenotyping. And so here's what this looks like with the Spinnaker system. We're able to fill the stacks of hotels on the right with plates that have barcodes. These are either source plates that have colonies from the library that have been randomly spread for growth, or target plates that'll be the recipient of the array. So here, the robotic arm is introducing a target plate, and then it will go on and grab a source plate, and it has a barcode scanning step where it checks to make sure that we know exactly what plate's being picked to what. It removes the lids, puts in the source plate, and now it takes the picture, measures the height of the agar using acoustics, and then it goes along and starts picking all of the colonies that we prioritize for picking based on how likely they are to be peers, so they're not touching another colony, and how far apart they are from another colony. And so we aim for roughly 768 colonies from each source plate. And the reason for that is we can fill a target plate half full at 1536 density, and then we can go on and grab a different source plate and resume picking until we filled up an entire array of 1536. And of course, this will proceed through other plates. And as you can see, the room gets dark because it's nighttime, and it keeps going. And so by the time we come back the following day, we can pick roughly 10 plates at 1536 density. And so over the course of a couple of weeks, we can actually pick a total of 200,000 colonies. And these are again spread across plates at 1536 density, and we can do this ready process at this higher density. And then we can consolidate the desired clones in a final step. And this looks like this. So here we've taken the 1536 density arrays and put them at 384. And now we've given the PIXL a file that it knows, so what source colony will go into what target position. And so in this consolidation step, we're able to generate these arrays that we then want to archive and process further. And so what we did here is we took this library and we checked to see, first of all, from a random set of clones, what are the whole genome editing outcomes? And we used this method we developed called SCORE to look at the structural variants that formed at these sites. And so what we found was that SCORE did an excellent job of predicting which sites would likely form structural variants. As you can see here, we had 88 structural variants in this library that were identified, whereas the majority were edited correctly. And so this is an example of one gene that's particularly structural variant prone called IMA1. All of the target sites here are above the SCORE-SV threshold of 0.15, and you can see we do have a very high fraction of structural variants formed. And so this highlights an important concept that in a pooled editing assay, even though the barcode uniquely tags a guide donor, that barcode is going to be present in many independently edited cells. And each one could have either a correct edit or a structural variant form, and depending on the location, this could be at 50% of each. And so if you're assaying only the barcode in this pooled setting, you'll have a confounding effect of a large fraction of structural variants that make it essentially impossible to know the true phenotype of that variant. Whereas if you can isolate these as clones and validate the edits, then you know that you have a correct phenotype that you're interrogating for that barcode. So we can do this across the whole library, and what we can appreciate is that with SCORE, we can prioritize which sites really need whole genome sequencing to validate that the correct edits have been made. So above 0.15, we would recommend that validation occur. Below 0.15, the entire approach could be done in a pooled setting, although it's also beneficial to have clones for those for downstream functional analyses. So what we do is we take the pools that we generated from the consolidated collection by washing those colonies back into a single pool, and we can grow them in a high throughput phenotyping system that we developed with TCAN using the EVO liquid handler along with plate readers. And so what this does is it shakes 48 well plates that have the libraries. In this case, they're in the top row. And when they reach the saturated OD that we specify, we will transfer an aliquot to some other growth condition. And we'll continue to do this down the plate as it grows. But importantly, we also save an aliquot, the rest of the sample, on a cold block that allows us to isolate genomic DNA to sequence the barcodes that we've identified. And so this system, much like the PIXL spinnaker, operates 24/7, and we can assay the entire growth across a plate using this system. So that's an example of how that works. And what the data we get from it, we get, first of all, the growth curves to show us the level of selection pressure that we've put in each condition relative to the YPD control. And so what you can see here are 6 passages starting with the first row of a YPD growth, that's 0 generations, and you can appreciate that we're getting quite a range of growth defect here depending on the condition. And we save aliquots at each time point. And we can do up to 24 conditions in a single run with this system. And now what we have here are the examples of the data we get. So each point is a barcode that we've isolated. And the y-axis here is the fold change of that barcode after 20 generations relative to the time 0. And on the x-axis, we see the genomic location of each variant relative to each gene here. So these are genes where we found that multiple causal variants resided in them. And these were all missense variants in this case. Across the whole data set, we've identified 398 causal variants, and this is across 24 different conditions. And these include all types of variants, including missense, non-coding, and synonymous, although we are enriched for missense variants, and we are able to capture 2 previously identified missense variants as positive controls. And across the whole data set, we do see an enrichment for missense variants, but we see quite a few synonymous as well as non-coding hits as well. And the effect sizes for the missense variants are the highest, as we'd expect, but we do see that the non-coding and synonymous variants have an effect. So currently we're validating these hits through additional whole genome sequencing. One of these locations, again, is in this GPR1 G protein coupled receptor gene, where we see 2 variants that are initially looking like they're far apart, actually fold up together, near in contact with each other at the sugar binding site. And we can see some interesting trends where large sugars that have 2 or 3 saccharide groups tend to be disfavored by these variants in some conditions. And this makes sense with the strain from which these variants came. So in conclusion, we've defined the editing outcomes genome-wide from CRISPR Cas9 editing. This system is applicable to other forms of Cas editing, including Cas12a as well. And so what we know now is that the genome is mostly editable with high fidelity, but the particular regions undergo high levels of structural variant formation, and we can define what they are in a predictive fashion across the whole genome. We built a new system called Majestic 3.0 that enables the highest efficiency editing to date by combining 3 different approaches to enhance HDR. And we use this system to map causal variants across 16 strains from the yeast species. So with that, I'd like to acknowledge all the colleagues involved in this work, in particular, former colleague of mine, Justin Smith, as well as Sybil Vanesh and Shengdi Li, who did a lot of the work in collaboration. And of course, I'd like to acknowledge the robots from Singer Instruments and Thermo for the spinnaker arm, as well as the TCAN system from TCAN Genomics.
Thank you very much, Kevin, for that really fascinating introduction to your research. It's really inspiring to me and to the whole team here at Singer Instruments to hear about the work of our awesome customers like you. It's our mission at Singer Instruments to accelerate research for biologists who are making the world a better place. And fundamentally, we're doing this by designing quality instruments that are insanely easy and also fun to use. Instruments like PIXL and Rotor, the 2 robots behind Kevin's high-density arrays. And if you're interested in finding out more, please do join us for the live PIXL demo we have coming up next in our webinar series. You'll be getting some info about that straight to your inbox, so make sure you save your space. That's all we have time for today. Thank you very much for coming, for joining us. I hope you've enjoyed the session, and I look forward to seeing you soon.